It's a pleasure to be with everyone tonight to talk to you about some of the technologies that we're developing in my lab to diagnose and treat disorders in the nervous system and what's called neuromodulation devices. So for those who are not familiar with this field, neuromodulation is the targeted interrogation of the activity in the nervous system. And many of us know that the nervous system exchanges information between our brain and parts of the body through the spinal cord by electrical signals, charged ions. And so it's natural to use the electrical modality to interrogate the nervous system and to be able to understand how it functions, and when there's aberrant function, how to treat it. Electrical stimulation has been used for many decades for treating disorders or diagnosing disorders in humans because it's effective and it's safe. And one of the most popular examples in which neuromodulation devices are being used is in pain management, particularly for lower back pain and leg pain. And in this case, an implantable pulse generator that you see on this picture on the right is implanted on the back of the patient, connected with some wires to leads that deliver electrical pulses to the surface of the spinal cord. And these electrical pulses basically interject with the pain signal and prohibits its flow to the brain. And some example of the devices that are being implanted are shown over here on the left. These have dimensions of about 1 centimeter in width, and the contacts, which are typically made of biocompatible platinum material that delivers the electrical pulses, are of the order of a few millimeters. Now, the same type of devices has been used in trials in patients with spinal cord injury. And when one controls where the stimulation is delivered, at which contact or which combination of contacts and at what time, one is able to basically provoke or initiate excitation into the motor neurons to be able to move the muscles. And as such, patients who have been basically paralyzed due to spinal cord injury can now stand up and walk in what is referred to as assisted locomotion. And the example shown over here is from a trial that's done in Europe and Switzerland, but there are many centers also in the US that are working for similar outcomes. Now, in terms of brain implants, some of the most popular use for neuromodulation devices is for patients with movement disorders, such as with Parkinson's disease, where deep brain stimulation is used in order to limit a tremor in these patients. Another set of clinical trials is also targeted toward spinal cord injury, but in this case with brain implants. So this is for brain-machine interfaces. And the example we see right here comes from Johns Hopkins Applied Physics Lab, where a paralyzed patient has been implanted with 6 electrodes on both sides of the brain, and using his thoughts that are decoded by these implantable electrodes, using the electrical signals that they evoke, he's able to basically control robotic arms and to cut the piece of cake in front of himself and feed himself. So these are all, very promising results in the research clinical trials, as well as in the therapeutic applications that we have just discussed for for Parkinson's disease and also for the pain management. So today, what I'm going to do is talk a little bit about the types of signals that we aim to measure with these technologies, and summarize some recent efforts or state-of-the-art in neurotechnology, and then discuss UCSD's efforts for developing high-resolution neurotechnology to interface with both the brain and the spinal cord. And I'll walk you through some of the development efforts, the controls that we do, and look at the safety measures that we take before we translate these devices from the lab to the bedside. And then I'll show some of the applications in mapping the human brain using these devices. And so the fundamental unit that processes information in our brain is called a neuron, and we have about 80 billion of such neurons on average in our brain, and these form a quadrillion of synapses or connections between them. And the gold standard technique to be able to measure the changes in potentials across this neuron cell membrane is referred to as a patch clamp technique, which allows us basically to get very close to the surface of the cell and measure the small potential fluctuations that happen within the cell. And those potential fluctuations happen due to the movement of ions through what's called ion gate channels. And these ions are like salt ions, charged ions, basically, in the medium where we have in the tissue, basically, where we have all of these neurons. And dysfunction in these ion gate channels is usually correlated with neurodegenerative diseases. And so a lot of the technology aims at understanding how these small potential fluctuations regulate disease or normal function in the brain. Now, as I mentioned, the gold standard is to measure each cell to be able to deduce how these small changes coordinate function. And there are technologies that are being developed that will allow us to look at hundreds and potentially thousands of these cells in what's called brain on a chip type approach. We can do that. But in intact brains, this is really very hard to accomplish, especially for millions of neurons. And so the way we currently interrogate or record brain activity is generally using three types of electrodes. Ones that sit on the surface of the skull, and these measure low frequency oscillations that are associated basically with long wavelengths that can reach further above the skull. Another type of electrodes is electrocorticography electrodes that sit on the surface of the brain directly under the skull. Because they are closer to the cells, these can measure slightly higher frequencies and over distributed large areas of the brain typically. And then there are electrodes that are penetrating to the structure or the tissue of the brain, and those can get close to individual cells and therefore be able to pick up the activity from single neurons. And so those allow us to measure basically broadband activity, but because they penetrate through the tissue and they cause damage when they do so, we usually limit their implantation size to a very small area. So then the challenge in recording broadband activity from the brain to capture the full spectrum of this activity over extended areas is indeed this dilemma between how fine of the details that we need to measure and over how much of the brain we need to measure it. So if we want to do high resolution, as illustrated earlier, this is going to be in a very small area of the brain. If we want to do large area coverage, we'll spread these measuring contacts over larger spacing, and therefore the resolution is lost. And this is limited by the number of channels that these electrodes usually are composed of, which is typically a few tens or hundreds. So then what we are doing at UCSD is basically looking at advances in the display industry and exploiting these advances to employ the new technology in mapping activity from the brain or the spinal cord. And those advances allowed us to have the high resolution displays that we all benefit from, and you're watching me through today. And it has advanced to a point where it's flexible and foldable so that it can basically confirm to the tissue, whether it's the surface of a brain or a cylindrical nerve or spinal cord. But there are more issues than just the number of channels that we need to account for when we think about interfacing with the nervous system. And one of these issues is the mechanical mismatch between the measurement tools and the tissue itself. And so we need to make sure that the measurement tool is soft so that it can be compliant to the movements and conformal to the curvature of the tissue. It doesn't have to be disruptive to the tissue, so it needs to have minimal size. Also, when it operates, it needs to produce minimal amount of heat so that it doesn't raise the temperature to dangerous levels. So usually you want to limit the temperature rise below 1 degree C above body temperature. It needs to consume minimal amount of power so that the battery or the power regulation unit is going to be small, and needs to operate with a high data bandwidth and transfer rate in real time so that we can basically assess and control prosthetics, for instance, in real time. So over the next few slides, we'll talk about some of the state-of-the-art technologies that are being used in research and then clinically, and then move on to the UCSD technology. So this is one example in which very small shanks, looks like threads or fibers that have multiple contacts on them, can be implanted in the tissue of the brain. And many of you might be familiar with the efforts at Neuralink, where they use an advanced robot to implant those threads by avoiding the blood vessels. And with this approach, they can go to several thousands of contacts implanted in regions of a millimeter scale. Now, these are being used in animal trials, and soon enough, we might be seeing them being used in human trials. Now, clinically, some of the most advances come from technologies that we already use for diagnostic purposes, like in epilepsy monitoring, for example. Clinicians use a grid that looks like the one that you see here on the lower left, which has a lot of platinum contacts, up to 256 channels usually, that are spaced by a few millimeters apart. So the lab of Eddie Chang at UCSF have really exploited this technology to advance the field of prosthetics, or in particular speech prosthesis, for a hand. Basically, what they have done is recorded the neural activity that's associated with the production of sounds by monitoring the kinematics of the mouth movement. And then using decoders, they were able to, just using the neural signals, predict what the patient is trying to say without moving his mouth. And this is an effort, that's led at UCSF, but also significant advances are being done at many other labs in the US and other places. So this is a picture of a typical grid that's used in these trials, and it's built usually on 0.5 millimeter thick silicone layers. The platinum contacts are usually about a millimeter in diameter, and they are about 4 millimeters spaced. Now, to put this in perspective, some of what we believe are computational units in the brain, the cortical columns, are of the order of a millimeter in diameter or width, and they are, of course, stacked very close to each other. So clearly, these clinical grids, while they have been very useful in treating patients, they undersample the brain activity. And if we want to make them smaller, these platinum contacts, the recording reliability becomes compromised. So we resort to advances in material science to allow us to make small contacts that can be stacked very close to each other, and they display low noise or higher fidelity in recording these brain signals. And at UCSD, my lab is specialized mainly in this interface, in making these very small contacts that can record reliably from the surface of the brain. And so I'll show you some of the results that contrast basically our new technology with the clinical electrodes in the next few slides. So this is a video recording of one clinical electrode placed on the surface of the brain of a patient. And you can see that this electrode, because it's built on slightly thick silicone layers, it doesn't conform very well or stick very well to the cortical surface. In some cases, it has to be cut, or the surgeons have to apply some pressure so that they could record reliably from the surface of the brain. This video shows, on the other hand, our multi-thousand channel grids. In this particular case, it's a 1024 channel electrode that's built on 10 micrometer thick biocompatible paraline material that allows it to be conformal to the surface of the brain, and it's also compliant to the brain pulsation. So with this conformality and compliance, there is really no movement of the contacts with respect to the tissue, and we can record with high fidelity from the brain surface. So first, we assess the functionality and the safety of these devices in animals, and before we translate them to be used in the operating room. So here we look at a well-established model, which is the Whisker barrel model that allows us basically to record activity in an organized structure of the brain by stimulating individual whiskers on the rodent. So in this particular case, we can see on the lower right, on the lower left, our grid placed on this barrel cortex. The spacing between the contacts is about 150 micrometers, and that's basically about the width or twice the width of the human hair, and it's much smaller than an individual cortical column. So now when we do targeted air puffs on individual whiskers, we can record the activity on these 1,000 channels and see that a subset of the channels have large amplitudes measured, and there's a time delay between some of the channels and the responses on the other channels. So there's some wave propagation. Individual whisker stimulation evokes activity in specific locations on the grid as opposed to others. So using this information now, we can basically look at the functional maps for each whisker or different organ of the rat and localize the functional boundaries that correlate with that organ. And we can see here, because of the 0.15 millimeter scale or 150 micrometer scale, these functional boundaries now can be delineated at sub-millimeter resolution. So to make sure that the results are scientifically valid, we'll have to go through multiple experiments, and we see reproducibility that will give us confidence in the results that we are seeing. And then for this fourth subject right here, we did post-mortem immunohistochemical analysis. And with a particular type of staining, we are able to see the organized barrel structure on the surface of the brain. So once we overlay our functional maps over that well-known procedure of staining, we found that the functional maps do correlate with the post-mortem histology. So that means, with a single electrode, we can really map the activity in the brain at a very high resolution. We don't need to sacrifice the animal to be able to tell where these structures are and correlate the responses with what we are measuring. And we don't need to do this serially across the surface of the brain. We have a larger coverage through the grid that allows us to get the maps that we have shared. And so in other cases, we implant these animals for longer time to look at the stability of the material in tissue. And we can also do histology to see if there are any changes or responses in the tissue due to the implant. Now, as mandated by international standards and the FDA, we need to do biocompatibility tests before we take these devices to be used in humans. So we contract with certified medical device companies that carry out independent tests about the biocompatibility. So once the device passes these tests, and it did pass for the UCSD multi-thousand channel platinum nanorod grids, we can then seek approval from an institutional review board to use these devices acutely in humans for a short duration time during the operation, or take those devices to the FDA for approval. Now, to get the devices to the operating room, we need to sterilize them. So we box them in traditional or typical sterilization boxes that are used in the hospital, and we go through the hospital sterilization. Then we seal our recording electronics in a leak-tight enclosure that's also sterile, so that we can separate the recording electronics from the tip of the grid that's going to be placed on the patient's brain. And then we do one more set of tests after this packaging procedure. Once the device passes the test, we can move on to generate or do our procedure in recording from the brain. Now, some of the
experiments that I'm going to show you next are done in collaboration with Dr. Ahmed Raslan at Oregon Health and Sciences University in Portland. So one of the tests that's typically done at the onset of the neurosurgery is to localize the different brain region to what's called the central sulcus. And this is the basically sulcus that separates the motor from sensory regions in the brain. And so a clinical grid, similar to the one that I showed you earlier, is applied to the surface of the brain. The patient has contacts on their hands or legs, and peripheral nerves are stimulated. With this peripheral nerve stimulation, some signals are evoked across this boundary, and if these signals basically transfer from the sensory to the motor cortex, they flip their polarity from being negative to being positive, for example. At the region where they flip the polarity, we know that that's the functional central sulcus. Now, in this particular patient, when we apply our grid, the results, of course, agree with the clinical grid, but we can map the curvilinear nature of this boundary due to the 1 millimeter resolution that we have. In fact, in this particular case, because the patient had a lesion, this boundary has been pushed from the anatomical boundary, which is defined by this large vein right here, to what you see as a dashed line as a functional boundary. Right, the brain reorganizes when one have a lesion. So the only way to know about this reorganization is to do these electrical recordings that we are doing here. So in some cases, the patients are awake, and our neurosurgery residents can instruct them or engage them in behavioral experiments. So the lab also develops hardware that could be placed on the hands of patients, for example, to track their movements and time lock it to the recordings from the brain. And so in one particular case, we've done 2 experiments with a patient in which we placed vibrotactile stimulators on the tips of their fingers and recorded the activity, and align that activity to the mapped functional boundary that we had before. And we see discrete correlates for each finger on top of the grid. And this is basically an overlay of the functional maps on the picture of the grid on top of the brain's patient, where we can see the distribution for the correlates in the brain for each finger. These are the sensory correlates. In another experiment, we asked the patient to close their fist, and then we were able to see the evolution of waves that bounce back and forth between the motor and the sensory region to coordinate this movement. And with the resolution that's afforded with these grids, now we can construct brain waves at 100 times higher spatial resolution than what have been achieved before, or look at brain activity in terms of waves, as we can see right here. This is basically during the preparation of the movement. We observed the brain waves originating from the sensory toward the motor cortex. And then when the movement is completed, we can see that this wave has flipped. And we have a time sequence of this movement throughout. Now, in other cases where there is a diseased tissue, like in epilepsy patients, a lot of times to map the diseased tissue, electrical stimulation is applied to the surface of the brain, while the clinical team is observing behavioral changes in the patient in response to stimulation, like speech slur, for example. Now, traditionally, this is done without recording because of the obstruction of the recording grid on the surface of the brain. Because our grids are thin and you can pattern holes in them due to the resolution of making the contacts, we have a lot of space to do other things like making via holes. The via holes are originally intended to perfuse cerebrospinal fluid from underneath the grid so that the grid maintains a good contact with the surface of the brain. But in this case, we can also apply electrical stimulation through these holes while we are recording, so that we can look at
the disruption of the epileptic waves throughout the stimulation procedure, and even correlate that with behavioral changes in the patient. So this shows basically one trial right here, and there are many trials that the clinical team do, in which we have first a stimulation artifact, and then a few seconds later we have basically a lot of discharges. Some of those discharges display a wave-like characteristic. So I'm going to show you next the evolution of these waves. In this case, without stimulation, we have a reconstruction of the patient's brain from fMRI and structural MRI images. And this is the location of the lesion that the MRI had identified. Now we relay on top basically patterns of the discharges. So when you see a black dot, that means there's an epileptic discharge happening there. And so what we saw actually when we have spontaneous discharges without any electrical stimulation, that the discharges start at the top left, top right in this particular image, and then spreads across the cortex and then exits from the other side. Now, in the case where bipolar stimulation is applied, and we will see in a second, the stimulation is applied, we have a large artifact initially, and this happens in the duration of milliseconds. And then we start to see discharges really originating from the stimulation location. And there would be still discharges happening in different parts of the grid, but we wouldn't have anymore the propagation of the signal from this point on the grid to the other point. Right, so we can see that we have persistent discharges in the stimulation location, and after a few seconds, that's the location where the discharges really fade away. Right, so what this tells us basically about epilepsy, with these trials, we cannot form a hypothesis yet about the different distinct nodes or the network in epilepsy. But this shows us basically that now we can really measure these waves, and we can measure these nodes and interrogate them by electrical stimulation at a resolution that may one day help us preserve some of the eloquent tissue in patients, or be able to stimulate with lower amounts of currents so that there are no side effects for patients that suffer from this disease. Now, in other advances, we can make custom-made or patient custom-made grids. In this case, this is a tumor resection case in a patient's brain, where we were told or sent the MRI images of the patients so that we can design our grid to have the flap that could be removed, and then resection could happen inside the grid while we are recording, and then this flap could be placed back, and we measure the electrical activity after resection. So this will allow us to basically track the evolution of function and how it changes during the resection, that we hope one day will help in having better patient outcomes from these surgeries. The lower right video shows exactly the approach that I'm describing, where the neurosurgeon is resecting the tumor, and then around the resection area, we have the grid recording throughout the whole time. So what we're trying to do is basically make this approach not only accurate and therapeutically beneficial, we'd like also to help in advancing the technology so that it's more comfortable during the monitoring procedure. For example, in epilepsy monitoring, the patient can stay for up to 2 weeks in the monitoring unit with electrodes implanted in their brain, and there's a lot of wires that are connected to these implants that are cumbersome and it could hurt the patient when they are trying to change clothes and so on. So what we're trying to do is connect our high resolution grids to a wireless radio implants that will allow us to transmit all of this data wirelessly without having to worry about the burden of wires. And we should be able to do that with all of the electronic advances that we have nowadays. And it's just a matter of adapting these advances safely to be used in humans. Another set of application would be to use these devices for brain-machine interfaces in patients with spinal cord injury. And we started discussions and collaborations with colleagues who are leading this field, whether it's in motor or speech prosthesis. So in the past half an hour or so, what I hope to have given you an update on implantable neuromodulation devices, an introduction to UCSD's emerging technology for interfacing with the brain and the spinal cord. I've showed you how this technology could be beneficial in animals and how we translate this technology at different steps to take it to the bedside, and then some case examples in diagnosing disease, in this particular case, epilepsy, as well as one example of recording brainwaves during movements or hand grasp experiment. So this work is done with in a collaboration with multiple excellent labs at UCSD and outside UCSD, in particular the lab of Ahmad Raslan, as I mentioned earlier, and Sidney Cash at Mass General Hospital, a number of talented students and postdocs, engineers and neuroscientists at UCSD, and also neurosurgery residents who have helped in translating those devices from the engineering school to the medical school. The work was initially supported by the Center for Brain Activity Mapping at UCSD, and once we collected some pilot data, we went to the NSF and NIH and applied for grants, and those grants were awarded. So the work is currently funded by a grant from the NSF and a grant from the NIH. We've made use of the Nano3 facilities at UCSD, which is part of an NSF-supported national nanotechnology infrastructure. And we've made use of the Department of Energy user facility at Los Alamos, where I did my postdoc before, and where some of my students have made parts of the technology when we started. So with this, I'd like to thank you again for your attention, and I look forward to discussions and to the questions that you might have on this or other aspects that you wonder about for neuromodulation. Thank you.
Shadi, thank you very much. That was a really tour de force going through some of the technology that you've been working on and how far you've come and how far you have to go, though, still. Let's talk a little about the science, because there are a couple of questions about what you're actually physically doing with this high-resolution grid. So before I go to the question, so what is the finest resolution that you've been able to have in terms of distance between electrodes?
Thanks for the question. So we can actually go as far as lithography allows us. And that's usually micron scale, where we can see things that are useful, where we can measure things that are useful. So in other trials I didn't show today, the smallest that we've done is 30 micrometers in between. So that's 30 over 1,000 of a millimeter, the smallest spacing that we've looked at. And we do see signatures of wave propagation, particularly in epileptic discharges, even at those scales.
So one of the questions was referring to one of your comments regarding a 4 millimeter distance at one point, but even with 30 microns. So their question is, how many neurons are in a given distance? Let's take the 30 microns. How many neurons are in that difference?
So neurons are arranged basically vertically in the cortex in different densities. At their highest density, they're in about a cube of 50 micron. In a cube with a size of about 50 microns. Now, in a single cortical column, which is like 1 millimeter by 1 millimeter, one have on average about 10,000 neurons. So basically in a 4 by 4 millimeter, which is the clinical grid, or this is actually a research clinical grid. Clinical practice uses slightly even larger separation. We're looking at a few 100,000 to 1 million of neurons. And as discussed in one of the slides, what we are looking at is basically the cumulative activity of these cells, right? So it's a regional activity rather than individual activity. To know about individual cellular activity, we need to get close to the cell and record individual cells.
Yeah, so this is a key question. You are looking at regional waves of activity as opposed to the individual neurons functioning, which is relevant to the next question, but I think you've in part started to answer this already. They're saying the measurement seems to be across a two-dimensional surface, and that is the case. Your grid is a two-dimensional surface. But the brain is, of course, three-dimensional. So there are connections that are going up and down, and the synaptic connections and so on. So how does this impact your understanding of what's happening here?
This provides us a piece of the picture, and this should be complemented with the recordings from depth along the depth of the brain. And so, we're doing surface electrodes and some high density depth electrodes, and other colleagues use depth electrodes in order to record the activity, stimulate, and try to understand how different regions of the brain work together. What the goal of the work that I just showed is basically to come up with a technology that will allow us to interface with the brain in a minimally destructive or a minimally invasive way without having to rupture any connections between neurons or the intact tissue of the brain itself. Unless it's diseased, we have to do that. Then, in my opinion, it's okay to go to depth electrodes. But if we want to preserve function and interrogate that function from the surface, we would be causing minimal damage, basically. So the short answer basically is that this doesn't give us a comprehensive understanding. It tells us a picture, like part of the picture, what's going on. We've shown that that picture is sufficient enough in order to decode speech, for example, decode movements, so we can use it for brain-machine interfaces. And some other labs have used it for therapeutic applications for stimulation, like to encode sensation, for example, and it's been effective. And that's why we're using that type of technology.
This reminds me, as you say, it's just a picture. It's not everything, but it's a sampling of this. And for a comparison, it is remarkable how much we have learned by not even opening up the skull, but just doing EEG recordings from people, which are even at a further distance from the brain, much cruder, and we can learn a great deal from that. And so now by getting down at this level, you presumably can learn much more. I did a calculation.
Localize the activity better, basically, right? So things don't overlap. So get better spatial resolution.
Yeah. And to express the magnitude of this, I did a calculation that probably a lot of other people have done, and I may have done it incorrectly, but I think the premise will be there. As you noted, there, with 80 billion neurons and all of their connections, there are a quadrillion synapses. So what does a quadrillion mean? And if I've calculated this correctly, that's equivalent to 1,000 terabytes of information if you considered it just to be bits. And so, some of you might have a computer where you've gotten it up to a terabyte of memory. You think that's incredible. A single human brain, 1,000 terabytes of information going on, and you're extracting part of that story with these grids.
And it's working at such a low energy efficient, and doing multiple processes at the same time that has developed over hundreds of thousands of years.
Yeah, I'm going to jump now to another set of questions because several people asked whether this could be used for certain things. And I think this is going to be an opportunity for you to say, we haven't done that yet, but that might be possible. But let me just go through some of the things. So one person asked, Can this technology be applied to blindness?
Indeed, there are actually clinical trials in which patients have been implanted with electrodes in their visual cortex. And those electrodes take images from pictures that are decoded into electrical signals. And then those electrical signals are stimulated basically in the visual cortex to try to provide as good as possible of a picture of what's in the field of view. The electrodes that have been used are more penetrating so far rather than placed on the surface. There are about 100 electrodes that I know of that have been in that implant. So one could get a perception of the shapes of the objects in the field of view, but not a full detailed picture yet. But yes, these technologies are being applied in this domain. There are labs around the world doing this, most notably in Australia. There are a lot of relevant clinical trials.
So do you have any sense of how far that's gotten? I mean, are there people who in these trials who were completely blind and can now make out shapes in their environment? And
well, I can speak of a study that was published in Science in December of 2020. And in that study, basically about 1,024 channel grids, which is 16 by 64 of these Utah arrays, I probably showed one of the pictures in my slides, were implanted in a non-human primate. And the non-human primate was completely blind, but with training, they were able to assess that the non-human primate is able to know the shapes of the objects in his trajectory. And so they are able basically to restore some notions of sight for this NHP.
Yeah, amazing. Thanks. So are there potential uses of this research to reverse chemo-induced peripheral neuropathy in cancer patients?
Look, it's possible. I don't have direct knowledge in this field, but we know that electrical fields can affect molecule movement and the growth of nerves. So it's possible that one can induce or prohibit certain biophysical effects with the application of electrical or magnetic fields. But I don't have more knowledge in this area.
Yeah, actually, since if I could throw in a bit because of work that I've done in peripheral nerve research some years ago, and because I am a survivor of cancer, having suffered from chemo-induced peripheral neuropathy, I think the neuropathy is usually basically think of it as an attack on the nerve connections between, for example, your finger and your spinal cord. So if that nerve has been knocked out, putting electrodes in the brain isn't going to help you to feel something at that end. You need to somehow replace that neuron and that connection if it's been destroyed.
Right. But, one could think about wearing a device that can perform the sensation and
Yes.
transfer the signal to the brain to encode the sensation. So things of this nature are being developed and tried right now. And for the patient that I showed was treated in Johns Hopkins University, they have that protocol basically of encoding sensations back from the robotic arms.
Okay, great, thank you. And then someone asked, with neuromodulation, could neuromodulation be used in a cerebral palsy patient so they could function normally? Is that a possibility?
Definitely it is. And, I'm familiar with the literature in the area that in the past, deep brain stimulation has been used effectively. And, the devices have advanced so that one can, wherever one is able to do functional mapping and targeting the appropriate anatomical regions in the brain in order to mitigate the disease, they are doing that right now. Obviously, for the pediatric population, not all children can tolerate these procedures. So there are, certain age limitations that would enforce inclusion or not in some of these trials.
Thank you. Still a couple of questions still on the possible uses. So this person is asking if all the person's nerves from the neck on one side were cut due to an accident and amputation was suggested, is there a way a prosthesis could be attached to be able to move the prosthesis with neuromodulation?
Indeed, yes. And, that's one of the goals that our technology is aiming to, but there is a larger initiative actually in the country. There's a program that was announced in late last year called Bridge the Gap that's funded by DARPA. And that's basically aiming to bridge the gap through the spinal cord between the brain and different parts of the body. And that's for both, recording and stimulation. That is reading signals from the peripheries and sending signals to the peripheries or to prosthetics.
Thanks. And so the next one is about Parkinson's disease. So they understand the disease is a result of the destruction of dopamine-producing neurons in the substantia nigra. So they were curious as to how the brain implant would work in that case. Does it slow down the destruction of those neurons at all, or is it more of a muting of nervous signals from the brain to the limbs?
It's obstruction of the neural signals from the diseased part of the brain to the limbs. It may have an influence on slowing down the disease, but the direct influence is really electrical, and that is to electrically disconnect the brain from the limbs.
Okay, thank you. And if I can take a moment, so talking about the stimulation of the brain, I know that one approach to studying epilepsy is to repeatedly stimulate the same place at a low level. And over time, if this is done appropriately, you can actually produce a seizure as a result, like an epileptic seizure as a result of that stimulation. The brain changes. It, in effect, learns from the repeated stimulation. So this becomes somewhat of an ethical question then, in terms of what you might be doing with brain stimulation. Is there a risk of this, and how would we protect against that?
I think, with like many things in our life, there is already risk associated with these implants, but there's always a balance between the benefit and the risk, and the benefit in most of these studies is higher. Now, with respect to the brain being plastic and changes over time and learning about basically that stimulation is muting my activity in this region, so I would rewire in order to basically bypass the stimulation. That's where the high resolution grids or the higher channel count might help, because then one would be able to more locally stimulate in specific regions and record from all of the surrounding region at high resolution. So if there are neurological shifts, then one could mute basically the stimulation in the previous subset and stimulate still in a smaller subset, but in a different location in order to counteract this plasticity.
So this is a point where, for a moment, if we could veer a little bit into science fiction. One of the questions that we always think about as we look at new technologies is to really be clear-eyed about what is possible and what is impossible, where things might be going. And before I describe this, are you familiar with a novel by Michael Crichton called Terminal Man?
I have not read it before.
Well, actually, given what you're doing, you should read it. I'm not saying it's scientifically accurate, but
okay.
But I'll tell you what the premise is. Somebody has epilepsy, and they decide to implant some electrodes in their brain to stimulate in appropriate ways to inhibit the seizures, so they won't have these seizures. What I am troubled by, I'll just go on the record in case anybody else has read that novel, is that this is a story in which the person who has epileptic seizures, the seizure, when they are unconscious, results in them targeting their attacks on people. So they actually physically go and attack people. And to my knowledge, that is not an epileptic seizure. Epileptic seizures are not targeted, they're unconscious and without that kind of connection. However, is there a risk of somebody having based on the stimulation starts to stimulate the wrong area and somebody could go crazy. Is this something that we should worry about?
Well, it's a very good question to ask and think about, but the likelihood of that happening is probably very low. It's well known where basically the lesions for epilepsy happen, and those are typically on the temporal lobe on the side of the brain, a little bit further away from more decision-making regions in the brain, the regions that affect the personality and the person's emotions. So, my first answer is that would be less likely, especially as we make the technology more target technology with having the higher resolution that we were speaking about. At that point, side effects are things that we are working to minimize. For example, now in some of the epilepsy implants, some people hear ringing during stimulation, and that's because of the gross stimulation of a larger volume of tissue. Then by having smaller contacts or stimulating smaller regions of the brain, one would want to minimize these types of side effects. So it's for a different reason than what you were thinking about, but the side effects are, of course, a very important consideration. That are usually taken into account in the design of the clinical trial or the design of the experimental study.
Thank you. So, and actually, well, since I veered into the science fiction piece, you're recording from the brain. You're recording electrical activity of neurons. So is this technology going to someday allow us to read someone's mind?
Maybe the intent to do some things, but get a comprehensive picture of all the things that are going in the brain maybe is several decades far from us. But we are doing already decoding for intents for movement, for example, or decision-based thoughts. We can decode this. Now, going deeper into levels, that's what's happening, at a deeper level in our thinking. I'm not sure the science is there yet, even though we might have like a mighty technology. Aligning it together with the science is still going to be a challenge that will take a few decades, in my opinion.
Yeah, I mean, so you may know that there are conversations about this sort of thing with functional magnetic resonance imaging, where people, for example, are theoretically able to tell whether somebody is lying or not. Is that
in that case, one is looking at some markers, that it's not looking at the detailed picture of the thought.
Yeah.
And I mean, there are many startups right now that are looking at what's called like speechless communication. That is, we get two brain implants, but we don't talk with each other. We don't even show any gestures, but our thoughts can be transferred between our brains. And is there some merit to this? There is scientific evidence that part of the things that we think about could be decoded and transferred. But how detailed that picture or how detailed this encoding is, it's not there to know. Like what I'm thinking right now, for example, how good are your questions, right? It will be hard for you to tell. I can tell you that it's very good, but would I have an implant that will tell you that in a few years? Maybe not. It will take longer time, especially the scientific basis needs to be sharpened. Even though the technology might get there, I don't think the science is there yet.
Thank you. So that actually leads directly to a question. We've somewhat anticipated it. In some ways, you've gone beyond the question. But so Elon Musk is a proponent of a brain implant for interaction with a computer. How effective is this at this point, or what can be done with the current technology? How far are we with that?
So that, they've basically made a few announcements, showed that their technology is really state of the art in terms of the number of channels or the number of neurons that one can record from the brain. And the two things that we know of that have been published is implants in mice, implants in pigs, and actually one third thing recently was posted through media is in a non-human primate monkey playing ping pong, I believe, with that particular implant. So, I think the advances there are marching toward an area where we can take brain signals to do things with our computers or with prosthetics. The field was already there. This company is taking the field to the next level in terms of how many cells one can read from. But there are, different scientific opinions or approaches to do this. Our approach is to read from multiple regions of the brain to get a complete picture. That approach that Neuralink is following is targeting smaller regions where things are really implanted through that structure of the brain. And so expanding that over larger cortical areas to really be able to decode sensory or motor information or other aspects of the brain is going to be a technological challenge. And it's going to be a medical challenge as well.
Yeah, I mean, I think all of us at various points when we use computers or our phones to look up information have the thought has passed our mind. I wish I could, write this more quickly, or I could get this information in the phone more quickly and get information back. We can do an amazing amount already, but the idea that we could somehow just think about a question and then have Google search for that question for us is very attractive, I think, to some people. So for others of us, it's worrisome what that means. Aren't we already attached to our computers enough? So let's see, I want to go next to a question just came up, which is a little bit different than some of the others about repairing some sort of physical connection or repairing a disease per se. But this one is asking about, can this technology be used to help people in correcting learning disabilities? Are there applications in that way that you've seen?
Yeah, indeed. There are some studies that are ongoing right now. For example, the studies are typically made on patients that already have other types of disorders. For example, epilepsy patients, in some of them, they have issues with memory. And then electrical stimulation has been shown to improve their memory over the baseline using such implants. So in normal people, it hasn't been tried, right? So the barrier to implant neuromodulation devices in normal people is high. But in people with symptoms who have already implanted devices to treat their symptoms, studies indicate that we can enhance the learning experience by electrical stimulation.
Amazing, yeah, thank you. This person didn't so much make this a question, but it's a statement that I think you might want to address. They said their biggest concern in terms of ethics is the long-term safety of these devices in terms of biocompatibility of implants. So what are your thoughts on that?
Yeah, and this is, very important. And, we do it in a way that we think we might get those implants at one point. So that's the level of care that we have when we do this. So in all of the devices that I know of, all of the devices have to be implanted in animals and to be in a large animal model, and to demonstrate that they are biocompatible for the life of the implant. Once that bar is passed, then the device could be moved toward clinical trials to be used in humans. And so the stability and the biocompatibility are some of the biggest concerns, and that's why we have multiple levels of checkpoints from the institution through independent contractors to get to the FDA that will review all of this information and make sure that we addressed all of these concerns before we get clearance to put it in humans. So I think the concern is valid. Now, could the FDA do mistakes, or could the scientists do mistakes in this process? They could. Usually, the things that we do are peer-reviewed and have to be, agreed on by the larger community. We wouldn't get resources if our work is not reviewed to be of some credibility by our colleagues. So there are still, again, a lot of checkpoints to be able to get there, and we have to go through all of these tests before we move on to humans.
Yeah, so there's an interesting tension here, and you mentioned this before, between risks that you see and what the potential benefit might be. And hopefully we have both science thinking about that balance and the individual making his or her choice about whether they want the implant. So, for example, I would presume that most of us would not be excited about having a brain implant put in our head so that we could, perhaps discriminate a little bit more difference in color in our environment. We can already see fine, but we could discriminate a little bit more. That might not be worth the potential risk of biocompatibility issues or infection or anything else. But if somebody has lost sight because of an accident, the chance of being able to see again would push in favor of saying, I'm willing to take some of those risks.
Right, and this conversation is actually right now is very active on the national level, and that's why we involve patients in the design of our experiments, and we start to involve patients in our grants and in our study sections, so that we can get the patient's opinion. Last year in August, we've done a workshop that was funded by the NSF in order to look at the advances in implantable devices and the needs. And we had patients who speak at the beginning of our workshop, and they, of course, raised a lot of concerns and raised all good points that they need to be involved in the design of the research, and also asked for the FDA to give them the right of their voice to be heard in the decisions whether these devices could be cleared for their benefit or not. Because in some cases, it's the FDA is more strict than what the patients would want them to be.
So the patients are advocating for loosening up the rules.
Yes, for getting some of the implants.
Yeah. So, so what are your thoughts about that trade-off of people pushing for something that might not be ready? Because isn't there a risk if we go too fast that people will be harmed, and then we'll have to step back and slow down again?
Listen, for the particular disease that that issue was brought up, I'm not familiar. I don't think the FDA would loosen up, or us as scientists would loosen up the bar in order to get a technology that's half baked, basically to the patient. There are cases in which the patient's quality of life is so low that they are in a position where they favor being subjects for trials for some of these technologies. Now, it doesn't mean that we have to do any unsafe procedure. We should go through all of the safety marks and the animal experiments before we do that. But how it's a very thin boundary that could be moved to the right and to the left, based on multiple stakeholders. So I think the most important thing is to involve all stakeholders in the conversation, look at the pros and the cons, and make an educated decision that will eventually benefit patients.
It sounds very good that you're doing that, and the challenge here that I'm picturing is, even as you do your research studies, if you at some point want to study a potential remedy for blindness, somebody who's been blind, who had sight perhaps when they were younger, and because of an accident they're blind, they may be very attracted to the idea that this might work. So how do you, I don't know what you've done in your own studies or some of your colleagues, how do you negotiate that informed consent process so that somebody who sees the benefit will also see the risks of what they're going to choose to do here?
Well,
How do you deal with that?
that's a really good question, and I think in the consent form itself, we'll have to inform the patient about the risks of the study, and what are the benefits, and there is any benefit, particularly to the work that I was doing, which was mostly for research purposes in this phase. But usually the procedures have to be done in patients who could, on their own, make a good, faithful decision and consent to the study. They understand all of the risks and the benefits, and they're able to do so. For patients who cannot do that, they're usually excluded from the studies.
Yeah, so I mean, just for the record, and I think it might be worth talking about this a little bit, I think I'm probably pretty capable of understanding things like that. But for example, when I hit an end user license agreement or a car rental agreement, I am notorious for pretty much not reading those. So how do you ensure that the patient actually absorbed the risks and didn't over-evaluate the potential benefit?
Well, yeah, I think the regulatory body would enforce basically the procedures based on their complexity. So if it's something that's really complicated and involves a lot of risks and some benefits, then this procedure needs to happen several days or maybe weeks in advance. And they have to involve the patient, to make sure that they understand every step and every risk before involving them in the trial. For some simple experiments, this could be done the day before the surgery, but for things that could be life-changing, it has to be done many weeks ahead or at least many days ahead so that the patient and their family could understand the risks.
And that sounds very good and ideal, to have a conversation so that it becomes clear the patient doesn't just say, I read the document and I'm going to sign it, but they're able to relay exactly what they're trading off. They say, I understand there's this risk. That's very good. So I guess related to this, one of the participants this evening asked, have any experiments resulted in injury or permanent injury to patients?
Not that I know of, and yeah, not in our experiments. I'm not familiar. Usually we simulate, of course, the experiments before we do them in humans to make sure every part of the procedure is safe. But I'm not familiar with any such incidents. Usually we have a safety monitoring board that's associated with every experiment that involves humans, and we'll have to report our progress and our findings and our incidents directly to this safety monitoring board that involves clinicians usually. And this is independent from the study team. They have the power to stop the study and until things are basically safe to resume.
So I'm just thinking about the future of how this kind of technology might be rolled out. I'm thinking of the stimulation side. I can imagine a pretty well, actually, I can imagine somebody thinking this is a good idea. So let's say that I have I am concerned that I have gained too much weight, and I think that the best way to stop gaining so much weight would be to not eat as much. And independent of whether that really would work or not, but let's assume that's what I believed would be the case. Then if I had an electrode that I could use to stimulate my brain to tell me that I was full, so I don't feel hungry and I wouldn't eat as much, then that would help me achieve my goal. And that, I'm guessing, might be a relatively low-hanging fruit in terms of being able to do that technologically. You just have to find the right area of the brain to be able to stimulate it appropriately. But is that the first thing we should be doing? And so
indeed, there are studies already doing that,
oh,
showing
really?
that
Okay.
actually brain stimulation can help in controlling obesity, can
Yeah.
help in controlling depression. It's really depending on the situation of the patient and how far they're out of control.
So these are involved implanted electrodes into the human brain
Correct.
in order to do this.
Yes, mostly
And
by the brain stimulation.
yeah. So, you know, I'm just speaking off the top of my head here. It seems to me that this is a situation where I would want this to be somebody who was literally morbidly obese and their health was at risk. Because as you've noted, I mean, we're doing the best we can in the research to try and be sure it's safe before it's implanted in a human. But there are risks when you're doing something new. So
yeah, so this is basically piggybacking on technologies that have been shown to be safe and effective for many decades, but steering its application toward a new disease, a new application.
yeah, okay, good. I want to take a brief aside because I really appreciate the question, and this is one of the things that can be a side benefit of these programs, is that somebody who hasn't identified their name, but they told us something about themselves. They said, amazing presentation. I am an undergraduate neuroscience major, and I was wondering what kind of professional or academic steps you would recommend students to take if we want to work in neurotechnology.
Well, I would recommend that you continue doing what you're doing, is basically listen to as many talks as you can and to learn, and also to identify your likes so that you are more informed when you make your decision which area basically you want to specialize in and become your best. And of course, the best way to do that is to have a lot of data collection and know about the different pieces that fold into this story of this technology and its use, and then think about where you as a person can contribute most into these pieces. So I think, make sure not to rush going one direction or the other, rush in learning like a lot of things. But then after learning, make your decision which aspect you want to go deeper in.
I just thought of something we haven't done with our programs before, but might be interesting here. And I'm going to ask the question about something slightly different for the technology, which we may be there soon. What if somebody, had learned that they were at risk of developing Parkinson's disease, but did not yet have Parkinson's disease, and a design was developed so that you could implant in the brain an electrode that would stimulate in such a way that would forestall the development of Parkinson's disease. So my question for the audience is, how many of you, based on what you know of where we are, where this is going, you can use the Q&A, just write yes or no. How many of you would say, yes, I'd go for that if I knew I was at risk of Parkinson's disease in the next few years? And you could put no if you think I wouldn't put the electrode in. So let's see what happens for the Q&A that way. Right, so far no's are winning. I wish we were in person because we could actually ask people, and here it's not comfortable to do that where people are theoretically somewhat anonymous. But actually, the no's came out early, but the yeses are starting to show up as well. And then somebody quite correctly said, I would need much more information before saying yes or no, because they would need to go through that proper informed consent process. So this is, I think, one of the challenges of new technology. Is it's really exciting to think about what it will be down the line when it's all working perfectly and everybody just goes into the clinic. It's an inpatient or outpatient operation. It's really easy. But moving from zero to that step means risks along the way. We don't know whether some things will work. We don't know what things might be possibly problematic until we start doing it. So we have to be the risk takers in order to make that, to allow for that.
And one other thing we also would want to think about is if things work out after the implant and you have some reconstructuring or some reorganization of the nerve cells, and this happens, and it happened in the context of spinal cord injury, where electrical stimulation induced bridging basically across the damaged or severed nerves, so that you don't need electrical stimulation after several months of the implant. And the patient, actually, because I think this has been translated recently to humans, the patient have regained some of their function. So then, is it safe to remove the implant? And what is the impact on the tissue when the implant is removed? How safe it is to remove the implant as well? So not only putting it in, but also taking it out should also be thought about, very carefully, not to cause damage to what has already been repaired.
Yeah. So there's another question that is implied by some things I saw and that wondering about, and I appreciate this is putting on the spot a little bit, but that's my job. So, so if at some point the technology you're working on gets put in someone's head, and maybe it is doing the things it was intended to do, and so that's good, but it also does something else that was unintended and causes harm to that person, maybe even severe harm. Who is responsible for that? Are you responsible for that because you came up with the technology? Is the doctor who did the surgery to put it in the patient responsible? And yeah, so how should responsibility be allocated here?
I think we all should be responsible. And, we started by talking about involving everybody in the decision. That's, of course, based on the things that we know, but there are things that we don't know. So in general, I don't think anyone has to hold a responsibility if they unknowingly caused some harm and hold that responsibility on their own as a single person. I think all people involved in the study will hold the responsibility in that respect. And that's why I tend not to take single-minded decisions without talking to my colleagues, even in engineering, but also on the medical side, to make sure we covered all of the effects that could happen, before we do the implants. And, we've talked a little bit also about side effects today, right? That, there are side effects. And this is something that, we're carefully thinking about. And the community now, given that the technology is advancing, is thinking more about how to reduce these side effects and be more selective toward only the intended use. So my hope is that, as things develop, we are truly more selective to specific targets in the modulation, so that we don't have to worry about these other things, other side effects, or things that we could damage in the procedure.
Yeah. So if you think about what you're doing, and it sounds like this is something that's routinely part of what you do, trying to engage stakeholders and getting an understanding of what's important and what's not important and what will be helpful from the community. To what extent would you say that is a common approach in engineering? Do you feel like this is something that most of your colleagues do, or is that unusual?
I think most of my colleagues do that. I would say that most successful approaches do follow that type of model, where you involve the end user and you involve the user itself while you are designing the system. And so we do that routinely here, and we try to have in different sectors of our research.
My sense is that we've had many scientists talk in this series, but only a handful of them made it really clear this is an important part of what they do. And I want to applaud you for that and seeing that, because I think it's important, because to do otherwise risks not only your research but the research of others, because if things go wrong, then that's going to reflect on all researchers, not just what happened in your study. So that's I think I agree, it's important. That's why we have this series.
The real success is actually measured, by the beneficiary at the end. So if you don't hear them from the beginning to the end throughout the process, then we're not solving their problem. We're solving a problem that might be irrelevant.
There's another question here that's an angle that I hadn't thought about. I'm hoping you have. So the person says, how mechanically sensitive are these implants? If some future patient had one of these implants, to stimulate a particular area of the brain, then they were in a car crash or some injury that could shift the implant out of position and therefore stimulate the wrong area or do something. So what's that risk?
So it's actually a very good question. And this is usually thought about in the context of designing the implant. It's thought about in terms of natural movements. For example, if we have an implant in the spinal cord and then the IPG or the pulse generator is implanted somewhere on the chest, and then the patient or the person who is implanted that implant has different types of movements of their neck to the right, to the left, and twisting their back, and that leads to some extension. And usually the leads and the device has to comply to all these flex movements. In the case of accidents where things might break, one has really then to have taken into account the possibility of going in, removing the device safely, and replacing it, right, if things mechanically break down. But in terms of natural movements in the brain or the spine or the other parts of the peripheral nervous system, this has to be accounted in the design stage, and we do leave room for these mechanical perturbations. And that's one of the main reasons, actually, we make our devices on a very thin substrate so that it can comply to those movements without affecting the fidelity or the quality of the interface with the nervous system.
So that question and your answer then makes me wonder about another reason that things might change, and that's development. So is there any sense that you probably would that you would normally not want to put one of these devices in a young child for the long term, because as they develop, the shape of their brain's changing, the interactions between the neurons are changing remarkably. It may be that that it just won't be the right spot for a long time, and therefore you get back to the problem of having to go back in to do another surgery. Is that a conversation that is going on about the risks?
Well, I think that's probably known, and we have to have like a medical person or medical expertise to say it. But I think after a certain age, the structure and the size of the brain doesn't change much. Things rewire within the brain, but the overall, morphology and structure doesn't change much. And this happens, at a child's age. I should not give you numbers so that I wouldn't be quoted wrongly. But it's not, for basically too many years from one being born. So that has to be taken into account. That's why those devices are not put permanently into somebody who is basically their brain size is still developing.
Yeah. So if it seems that age is probably is as you say, and I'm not going to give the precise number either, is fairly young. But even if you ignore that, we do know that all of us who have had teenagers at home or remember ourselves as teenagers know the brain may not be physically changing in size, but it is changing in its connections. And so that depending on what you were treating, I could imagine many treatments that might be anticipated that that those areas of the brain that you're trying to stimulate or inhibit might be changing. And so you might need a plastic or a changeable array that's in the brain to be stimulating different areas.
Indeed, and maybe one other way to look at it is to have more, again, targeted regions to stimulate or record, where some of the electrodes are not activated for a long duration of time, and those will be implanted in closed loop systems. So let us say you stimulate to get a certain function, but you also measure the biomarkers of the effect. And then, if you're not getting the desired outcome, then you can change the location of the stimulation and read the biomarkers in order to adjust. So that's where having more contacts and higher channel density or counts is important.
Yeah, well, that's a theme that's been throughout this talk and your presentation. Is you are looking at both ends of this. You're interested in stimulating electrical activity of neurons, stimulating neurons, but also recording activity, and that ability to get feedback for what you're doing is key. So I think we'll need to wrap it up now because it's 7 o'clock, but I want to thank you for a really interesting, wide-ranging talk. We covered a lot of different potential outcomes here. I think you have a lot of people in the audience who are looking forward to new developments. They may not come fast enough, but I know you're working on it.
These were excellent questions and really deep thoughts. So I appreciate all the points that you have raised and the audience have raised.
We have a very good audience, so that's to be expected.
Yes.
So, thank you, Shadi. Have a good evening.
Thanks a lot. Have a good evening.